All right. Hello and welcome. Thank you all for joining our co-hosted webinar with IDEXX BioAnalytics and EditCo Bio. I'm Steve Smith, our Senior Product Manager at IDEXX with our cell health portfolio. And we love working with our scientific partners behind the scenes. So today we're really excited to share more about why partnerships like this are so important for us and for you all to help deliver the outstanding research results that we're all after. Today, you'll get a chance to hear why cell health is a cornerstone element of any cell-based research program, and also learn from Travis at EditCo how to bring some of the most cutting-edge techniques in biology today, like CRISPR gene editing, into your workflow. So our speakers today are Travis Hardcastle from EditCo Bio and Sarah Hansen from IDEXX. Both have impeccable academic credentials and accomplished scientific careers in the industry, and they're excited to talk about topics near and dear to their hearts today. So let's turn over to Sarah first to talk about cell health for today's advanced cellular models. Thanks, Sarah.
Awesome. Thanks for the introduction, Steve. As Steve said, I'm going to get started just talking about cell health, and then I'll turn this over to Travis in a little bit here. So for over 60 years, we know that cells have remained at the foundation of modern biomedical science research. And for that same amount of time, then pretty much, which I'll show you later, questions have been asked about the validity of studies utilizing cells. So for our webinar today, I'm going to start off with the goal of emphasizing 3 cell health diagnostics to enhance study validity. So the first is that cells should be clear of pathogens for proper study outcomes. And in this area, I'm going to include mycoplasma, bacteria, fungi, yeast, and viruses. Number 2, cells need to be verified by some molecular method to match the origin, and that is either the original cell line or the original patient donor. And 3, cells need to be determined to be genomically stable, especially when we talk about stem cells, which we'll get to a little later. So for step 1, I said cells should be clear of pathogens, but when evaluating cellular pathogen status, the source can be a little bit helpful when you're setting expectations. So for example, the cells that are shown here came from a vendor, and they come with the information that the cell line is positive for EBV. If the source is shared cells from other investigators, previously banked cells, or primary cells or human tumors, then further testing for human pathogens is going to be very essential. And this is for 3 main reasons, one of which is to protect your personnel that are working with the samples from the pathogens. 2 is to protect the research animals that are in the vivarium. And then the 3rd is to ensure research integrity. And this slide shows prevalence data of tested biologics for common human viruses. And as you can see here that EBV is the most common positive. This kind of makes sense when we think about the fact that of the human population having such a high prevalence rate of EBV. And then this is followed by HIV-1, HPV-16 and 18, and several other human herpes viruses. Likewise, your biologics and cell lines that have been passaged previously through mice should be tested for mouse pathogens. And this list is the most common ones that we tend to see. These agents are not only excluded from modern immunodeficient rodent colonies, but this is because they can cause severe adverse outcomes that are unrelated to the model itself. And this was shown really nicely here in this 2019 publication, comparing C. bovis status in a chronic myelomonocytic leukemia model in NSGS mice, where they found unexpected study changes in many parameters, including circulating platelets and neutrophils, as shown here, as well as spleen weight, and then failure of the model itself when they had C. bovis positive mice. So you can see that in these histology images here where the expected CMML blasts were lacking in the liver and the spleen. So this shows how this still very prevalent rodent pathogen can significantly alter the outcome of a study. And the same is true of human pathogens. There's recent evidence in the literature showing there can be reactivation of latent herpes viruses in cell culture And this can be a challenge in donor source cells. So this 2023 publication here shows in figure 1 just a quick review of the viral life cycle in humans, where following primary infection, you have long-term latency that can be then reactivated with particular environmental cues. And in figure 1D here, evidence of HHV6 reactivation from donor T cells. So again, these studies have huge value, and knowing that viral status of the cells is extremely important. Likewise, ensuring that cultured cells are negative for bacteria and fungi is very important. While some bacterial infections will be readily obvious through media color changes or visualization, not all of them are evident. So as shown here, this is a 2020 publication where this lab experienced bacterial contamination kind of widespread throughout the lab of cultures that they found was related to a spore-forming bacteria that originated in their DI water tap, in fact. So it was kind of spreading that throughout their cultures. And they found that these bacteria can alter cellular function and measured cytokines, which were common study parameters for them. So obviously created a big problem. And it's also important to remember that some bacterial populations can alter cellular behavior, both in vitro and in vivo. And this was a really good example showing that the proliferation of colorectal cancer cells is altered when they're co-cultured with Fusobacterium nucleatum. So it's really important to find these pathogens and ensure that your cells are clean of them. And then to complete any discussion on pathogen testing, it's important to talk about mycoplasma and remember that mycoplasma remains to be very problematic in cell culture. And it's one of the most frequent positive tests that we find. And I'm bringing up this 2015 publication here where this lab found mycoplasma in their samples. So they wanted to look and see how often has this been happening. They searched back into the archives at NCBI for mycoplasma reads within sequencing data, and then they compared these results to publications and then the number of times that each of those publications had been cited. So some of this data is shown here, and even if they limited their search to reads for the 4 most common mycoplasmas, they found published studies in Nature, Cell, and others. And out of these 18 publications summarized in this, they were cited over 2,000 times, with 2 of these publications being cited over 500 times each. They concluded overall contamination rates of anywhere from 15 to 70%. Depending on how you look, these numbers shift a little bit. So we know this is still quite prevalent, and we know that mycoplasma has serious effects on cellular function, so must be continued to be excluded from cell culture. So we've had mammalian cell culture since 1943, beginning with a mouse cell line, and we've had human cell lines in culture since 1951. And it was found shortly after this, and this publication that's shared here from 1974, this group began to recognize that there was something about the chromosomal pattern in one of their cell lines that appeared to be distinctly similar to that of HeLa cells. So this was shared again in 1974, 50 years ago, and contamination is still something that we're tackling today. This topic drew enough interest, actually, to result in a Rolling Stone news story, which I thought was quite interesting. It actually made it on the cover of Rolling Stone in 1976. But again, despite this, we're still seeing these issues today with problems of contamination and lack of cell line authentication. So cell line authentication through the use of STR profiles is the method that we use to confirm that the cells that you're using are authentic to their source. And this testing consists of short tandem repeat PCR profiles where sequence-specific PCR primers are used to target polymorphic microsatellite regions. These have anywhere from 2 to 6 base pair nucleotide repeats within them. And then the number of repeats at each specific marker is determined, then converted into an allele with a numerical value, and a profile is generated. So in the example that's shown here, at the marker shown, this cell line number 1, there is 2 and 4 repeats for each allele. But for another tested cell line, there's 3 and 6 repeats. So when these markers are then all put together in a large panel, a unique profile can be identified. And the identification method that we use follows the American National Standards Institute markers for consistency. So these can be compared across researchers. So here's a sample profile that was obtained from a submitted cell line. So this sample was submitted as NCI H1792. And according to the national standards, then these profiles definitely do not meet the 80% match criteria. So you can see in the center column here is the sample results. So the alleles that were read at each of those individual markers in that 16 marker profile. And then the reference profile for the cell line that this was intended to be is on the right. Anything that's in teal green here then is a mismatch. So clearly this is less than an 80% match. But when we evaluated this profile, this actually was found to have a 100% match with the Kasumi 6 cell line. So this is a true example of misidentification, in fact. Okay, and then lastly, the third point that I wanted to make is that I mentioned that genomic stability should be monitored, and this is particularly important when one is using stem cells. So as cells are cultured and manipulated, stress to these cells can result in copy number variants and mutations. And while duplications are the most common type, deletions can also occur. And now many of these have been identified and published, including the most common copy number variant, which is the 20q gain, which is shown in these publications here. And this has been shown to promote survival advantage in cells in vitro. So it kind of makes sense why this would most commonly be found in these. So identification of these cells that can exhibit the CNVs is important then to ensure overall cell health, as these may have negative downstream effects, as shown here in this 2021 publication. And since 2013, this has been a statement actually from the International Stem Cell Initiative that screening for genetic abnormalities is paramount to claiming that these cells are normal. So together with STEM Genomics, then we now offer a rapid screening, and this is done with digital PCR and provides evaluation of 24 copy number variant hotspots throughout the genome. And this publication down below, if you pull the reference, you can see all of the data, but basically in rows B through K, this is a summary of the published and known copy number variants that occur in these cells. And then it's put together here in the top in row A to show the assay that and the hotspots that are covered by the assay. So this assay has a rapid turnaround time and covers up to 93% of the published CNVs that are out there, and has a very straightforward analysis as well, and provides some data when you do find the CNVs of what that may mean for those cells. So with that preview into some foundations of cell health, I'm actually now going to turn this over to Travis to talk about how all of this applies in a practical setting.
Great, Sarah. Thanks so much for the informative presentation on cell health. And as Steve mentioned and as Sarah mentioned, my name is Travis Hardcastle, and I'm the Director of Product Management here at EditCo. So, who is EditCo? If you're in the genome engineering space or not, or we might be a new name to you. However, EditCo in its current form has actually been around since about 2012, but we were under the umbrella of Synthego. Earlier this year, the engineered cells and multi-guided reagents business spun out of CRISPR to form EditCo. And as part of that, we actually have quite a bit of a legacy of expertise in the genome engineering space, as seen by some of the numbers on the screen here. We've been responsible for well over 500,000 genome edits when taking into account our services and our CRISPR reagents portfolio at this point. Additionally, we've been cited in well over 700 publications when you take those same products and services into account as well that we brought over from Syntheco. But from a cell line engineering perspective, we fine-tuned our automated platform to have an average editing efficiency of around 80% for our knockout pools. And to date, we've worked with it's probably well over 600, probably closer to 800 cell lines at this point. This data is a bit outdated, but closer to 800 cell lines. Additionally, we worked with and collaborated with many of the top researchers in the world, places like St. Jude's, In Citro, and one of my favorites, MD Anderson, which is where I began my career in research, many moons ago at this point. So, with that said, I know that we probably have a wide range of sort of scientific disciplines on this call, and while CRISPR has been around for a decade now, I wanted to do just a quick level set to make sure that we're all aligned and on the same page for what CRISPR does and how it actually works. So let's go back to the beginning of the genome editing evolution with mouse engineering. In the 1980s, this was a point in the genetic engineering timeline that turned many genetic experiments into real-world sort of products and ideas, really changing the landscape of what was to become in the 1990s. In 1981, there was the first transgenic mouse. A transgenic animal is quite simply any animal that has had a gene from a foreign organism deliberately inserted into the genome. Today, generating transgenic animals for scientific experiments is quite common, using many modern genome editing methods. But for the history of genetically modified animals, the process first began with a research team led by Thomas Wagner at Ohio University in 1981. Wagner and his team transfected the gene of a rabbit into the mouse genome using a method now standard in genetics known as DNA microinjection. Their experiment paved the way for revolutionary transgenic animal experiments in many of the years to follow. Then, in the 1990s and early 2000s, came a discovery of the technology that I, as well as our host Steve, became very familiar with from our time at GE Healthcare DharmaCon, which later became Horizon Discovery, and that is RNAi. So RNAi was first discovered in the 90s, and throughout the early 2000s, made leaps forward where it made the clinical space using RNAi in therapy. Around the same time came along zinc finger nucleases, or ZFNs. So ZFNs are possibly the first sort of technique that, for highly targeted genome engineering. The discovery of zinc finger nucleases improved the effectiveness of gene targeting in several ways. Zinc fingers helped recognizing quotes DNA, making it possible to design ZFNs for a variety of different genomic targets, varying throughout many different types of cells and organisms. Each ZFN binds specifically to a set of 3 bases. It can be combined to recognize longer DNA sequences. Researchers have fused the DNA cutting enzyme FOLK1 to the ZFN DNA binding domain to create a sort of genomic scissor, sort of similar to what we see with CRISPR, which can cleave the DNA at a specified location, creating a double-stranded break in the DNA. If a repair template is co-transfected in the cell, then a fraction of the cells will undergo this homologous recombination, where the cell incorporates the template insert into the specific breakpoint. This new technique happened to promote the idea of backwards genetics versus a forward genetic approach. So with forward genetics, as many of you guys probably know, the scientists will generate random mutations looking for something significant, and then characterizing the gene they believe is responsible for the mutation. However, with backwards genetics, thanks to this gene isolating methods like ZFNs, we are now able to identify the exact gene causing the mutation and attack it specifically. So while ZFNs represented a revolutionary change in the way that we manipulate the genome, their DNA binding domains are notoriously hard to design, and the efficiency for cutting site of interest. The design process, therefore, can take months, if not yielding unsatisfactory results and off-target effects. So, Then came along in 2011 was talons. So in 2011, the generation of two of designer nucleases came to the form with talons, which recognizes a single nucleotide rather than the trinucleotide motif you see with ZFNs. Compared to ZFNs, talons benefit from the ease of design. Effective talons can be designed and manufactured in a few days and can be multiplexed with many on the order of hundreds of times. Talons appear to have fewer constraints than genome sites and can be easily targeted across many genomes. And talons were also found to be less off-target effects than ZFNs and are less cytotoxic to the host cells. While talons have clear advantages over ZFNs, they also exhibit a few disadvantages as well. They're significantly larger than ZFNs, so therefore they are harder to deliver and express in vivo. Along these lines, while knocking out genes with ZFNs and talons proved fairly straightforward, knocking in genes with this technology became very much complicated. So then, in 2012, Dr. Doudna and Amanda Charpentier introduced the world to CRISPR. Their teams elucidated the biochemical mechanism of CRISPR technology. By making precise targeted cuts in DNA, CRISPR ushered in an endless potential in areas of medicine, agricultural, biomaterials, and many, many more. So, how does CRISPR work? As I've heard it mentioned before by many of the pioneers of the CRISPR space, I like to equate this to the search, find, and replace function of a word processor. First, it's important to understand the components that make up the CRISPR mechanism. It consists of two components: the Cas9 nuclease, that is an enzyme that creates the double-stranded break in the DNA, and then the guide RNA, most commonly in the form of a single-stranded of RNA. However, there are some companies who offer this as a two-component form, in which the gene-specific CRISPR guide RNA is complex with the trace RNA. However, I'm not going to get into the pros and cons of each of these systems in this talk. Upon getting into the cell, the CRISPR-Cas9 complex is guided to this gene-specific target by the guide RNA, where it binds to its DNA target and allowing for the CRISPR-Cas9 or the Cas9 enzyme nuclease to perform the double-stranded break upon creating that double-stranded break. Which then we allow this natural sort of repair mechanism to be performed in an action called non-homologous enjoining, or NHEJ. Because this process is not perfect, oftentimes we end up with either insertions or deletions in the genome, and this is commonly referred to as indels. These indels often lead to disruptions of protein function and expression. So how does this occur? Well, we just have to go back to our basic biology class in which we learned about the central dogma of molecular biology. First, we know that DNA gets transcribed into RNA. That RNA then gets translated into protein, and this protein gets processed with post-translational modifications to fold into its active form. From there, you can perform functional assays to determine these protein readouts, right? This sort of this basic biology that we all learned back in grade school. But however, when these indels get introduced into the DNA, oftentimes this leads to an introduction of an early stop codon or disrupts the sequence in such a way that after the DNA gets transcribed into RNA and that RNA gets translated into protein and there goes this post-translational modification, it doesn't fold properly and leads to either a non-functional protein or one that is far less active when measured with a functional assay. It's important to note, though, that when this indel happens in a multiple of 3, it will lead to a frameshift mutation, which will sometimes allow for a truncated and partially active protein. So in this case, when screening for clones or any sort of edit in clonal cell lines or cell lines in general, it's important to isolate clones with an indel that is not a multiple over 3 to ensure complete protein disruption. So what about the other types of editing? We should also note that CRISPR can be used for creating specific SNVs for disease modeling, as well as larger inserts and tags, such as fluorophores, for examining protein localization. This process is a similar process to how proteins are knocked out. For example, it consists of transfecting with a Cas9 enzyme with a guide RNA. However, there's a third component that is added that allows for the precise repair of the DNA. Depending on the size of the insert, this can be either used as a short single-stranded DNA template that has homologous sort of sequences surrounding the precise knock-in that you want to insert, or plasmid that can be transfected into the cell, creating a much larger repair template. As with the short single-stranded DNA, the plasmid must contain the homologous regions surrounding the specific site in which you want to knock in the specific tag. So how can CRISPR be used by researchers such as yourself, and what applications can it be used for? Well, there's many different ways in which it can be used. Pathway analysis. So CRISPR enables us to systematically disrupt genes to understand their roles within biological pathways. This is crucial for dissecting complex biological processes and identifying key regulatory genes. Disease modeling. By introducing specific mutations associated with human diseases in the model cell lines with CRISPR, we can create an accurate disease model. This helps understand disease mechanisms and testing potential treatments. Screening and target identification. So with high throughput CRISPR screens, this allows for us to identify genes that are involved with various cellular functions, including those that could be potential drug targets. And lastly, one of the other ones I highlight on is target validation. Once potential therapeutic targets have been identified, CRISPR can be used to validate these targets and observing the effects of their disruption or modification in relevant cell types and animal models. The list really does go on, and this is not to be a sort of definitive or exhaustive list, but this is just some of the sort of key things that CRISPR is being used for by researchers these days. So, as I alluded to earlier, I mentioned a high-level overview of what EditCo is, but how do we go about doing what we do? So, at the core, EditCo is really focused on democratizing CRISPR. EditCo's mission is to make CRISPR technology accessible to everyone, from large-scale research institutes to small biotech startups to individual researchers like yourself. We aim to do this by offering an affordable and easy-to-use CRISPR kits, as well as selling engineering services. Along with our comprehensive technical support and customer support teams, we're removing those barriers that have traditionally limited the use of these powerful technologies such as CRISPR. For our engineered cell services, we achieve this through our ECLIPSE platform. ECLIPSE is really this groundbreaking CRISPR editing platform that aims to deliver the new gold standard in cell-based models so researchers leave no variant, gene, or mutation unstudied ever again. So what is the ECLIPSE platform, you asked? We just talked about that. Well, let's break it down to some of the key components. So chemistry. Our advanced chemistry through the expertise when we were a part of the EditCo ensures our precise and efficient genome editing software. Our powerful software tools and LIMS management system it manages and optimizes the entire editing process from front to back. Automation. Our automation increases the accuracy and efficiency while reducing the manual errors that are therefore improving our reproducibility within our edits from edit to edit that we'll talk about here in just a bit. And then bioinformatics. Our advanced bioinformatics teams and tools analyze and interpret a vast amount of data sets and form this what we like to call our CRISPR feedback loop, which really allows us to use machine learning algorithms to improve our guide designs and our precision and outcomes of our genome editing projects over time. So what you have here is really just the encompassment of all that. It's just a culmination of everything that we just talked about. It allows us to create the new sort of gold standard in cell-based models for researchers. It really allows us to create any edit, whether it's a knockout or knock-in in any cell line, whether it's immortalized primary or what's not listed down here, primary cells. It really allows researchers to take their cell lines from discovery and almost to the clinic. While we don't offer GMP grade cell lines, we do we can get you a lot closer to that with a lot of our editing capabilities. So what we have here is a bit of a sort of peek behind the curtain of our highly automated factory, which is based in Redwood City, California, or what I like to commonly call our cell nursery. The image on the right there is actually where all the outgrowth and expansion of our cells takes place. It's a highly automated sort of fashion in which it happens. Following for clonal projects, following single cell printing and clonal isolation, cells are actually moved around in this module in a large plate format, and images are actually taken every 2 to 3 days. We screen on average about 400 clones per project, and so as you can imagine, the data and images add up quite quickly whenever we take into account the number of projects we do. So I think at some point a huge shout out goes out to our data and software and automation teams for just designing such this intricate sort of system that's really high throughput and allows us to do this. But because of all this allows us to create sort of this high throughput automated platform that allows us to work with just about any cell line. We've been able to develop a system that allows us to test up to 200 optimized condition, and it allows us to identify the best condition for any new cell line that we work with. This has allowed us to work with those 800 plus cell lines that I spoke of previously, and allowed us to generate, just with our services alone, probably about 250,000 edits at this point, which is a big sort of accomplishment by our team. And here you can see a few examples of this optimization and how we've been able to generate this through this highly automated platform. So EMT6 is known to be a relatively easy cell line to work with. And as you can see, we've been able to identify probably well over 100 conditions that work with this specific cell line on the panel on the left. However, 5637 is a moderately difficult cell line to work with. But as you can see in the graph in the middle, we've been able to identify many optimized conditions that work well with this cell line. However, lastly, in the panel on the right there, the THP1 cells, these are notoriously difficult to work with. I know this from personal experience from my time in R&D, when they were giving me nightmares finding, the most optimal, the best editing conditions to work with. But because we've been able, to find the best conditions due to our highly automated platform, this is actually one of our most popular cell lines that customers come to us to work with, researchers come with us to work with to generate edits in, as we've been able to find this high success because in optimal conditions based off our platform and our abilities to streamline the process and find the best conditions to work with. But enough about EditCo and some of the things that we do. Let's focus a bit on more what this webinar is about, which is why we partnered with IDEXX and to create this sort of ecosystem for maintaining cell health and the importance it plays for our genome editing capabilities, particularly with iPS cells. So at EditCo, we offer a wide range of iPSC editing options, and this can come in the form of knockouts, SNVs, which is single nucleotide variants, as well as reporter and tag knock-ins insertions in the form of homozygous or heterozygous clones or pools, which are just heterozygous or heterogeneous population of cells. As you can see by the image on the right there, I actually went too far. I'll go back real quick. So yeah, this is just a quick example of some of the readouts that you get from our software, which is how we QC our edits. As you can see by the traces there with our sequencing, we have 100% sort of C to T conversion there at the specific base, and it's compared to the parentals on the lower bottom. And so this really provides us to provide accurate disease modeling with high speed and accuracy. But when it comes to small tags, as you can see, we can generate knock-in efficiencies of well over 90% of the pool population. And many of the common tags you're seeing here on the graph on the left with HA, whether it's a 3X tag, flag tag, a flag, S, a CBP, V5, or a MIC tag, we're having editing efficiencies in our pools that are well over 90% knock-in at the pool stage at this point. There's a bit of a fall-off, but still impressive knock-in of around 60% for our high-bit tag. And we also maintain and measure our pluripotency of our cells, as seen with the image on the right there, and to make sure that they do maintain this quality from our editing process and our editing platform. More importantly, we're able to insert these tags with such high editing efficiencies, these pools appear clonal. In this example, we knocked in this HA tag into a GAPDH gene, and we're able to achieve about a 98% knock-in efficiency for the pool. This can be visualized really well on the slide here if you look at the immunohistochemistry, as well as maintaining the pluripotency if you look at the SSEA4 marker there. But how does this relate to IDEXX and some of the activities we do with them? So for iPSCs, it mainly comes down to assessing the genomic stability of our cell lines. It's fantastic that we're able to edit these cells, but can we maintain the genomic stability in general outside of the edits that we're trying to introduce, even though we go through harsh conditions for clonal isolation and transfection? Here we show one of the services that we offer our customers through our partnership with IDEXX, which is assessing the genomic stability through the ICH or ICS digital readouts that Sarah spoke about earlier today, or earlier in the talk. But I wanted to take some time to go over a bit of a quick case study about the importance of this service that they offer and how it pertains to EditCo and some of the things that we're able to do on our platform. Stop responding. There we go. So what you see here is a typical pathway that is followed for generating an edited cell. A more detailed view of generating an edited cell in our pathway or on our platform. So there's editing strategies. What do we want to create, knock out or knock in? Do we want to generate a large transgene insertion or create an SNV? All of which can dictate the type of editing strategy that we want to employ. Then you can go on to guide designs. Depending on where you're targeting, you need to design several guides to identify the one that has the highest editing efficiency and therefore leading to the best chances of success. Optimization of transfection, we talked about that earlier. We have this 200-point optimization, and so we can pretty much find it and identify a protocol that works well for any cell line. Electroporation, this is where we deliver the CRISPR-Cas9 complex into the cells and allow the genome editing to take place. Then followed by pool analysis. So just within a few days after transfection, we can take the cells and allow the cells to recover. We can take samples of those cells and isolate genomic DNA and perform Sanger sequencing. Clonal isolation. This is where if the editing in the pool is high enough, we can go down to single cell printing, as we saw earlier with our automated platform, to take images and really allow the outgrowth to happen. But where IDEXX plays a critical role for EditCo is in our cell qualification testing and our QC methods, particularly for customers' supplied cells. But in this example, I'm going to talk about specifically our master banks and how we QC some of our master banks using services that IDEXX provides to us. So within EditCo, we have several iPSC cell lines that we allow our customers to choose from as control lines to start their editing with, or we can use their own cells if they want to ship them and provide them to us. But one of our most popular cell lines is our PGP1 cell line that's been around for several years now as a part of George Church's personal genome project. It's a cell line that we have a ton of experience with and we do many, many, many edits with. But that said, a few years ago, we had a complaint about one of these PGP1 cell lines, one of these edited clones that we provided our customer, and they came back to us and told us that it had this 20q abnormality that Sarah spoke about previously, and they discovered this using a BD, digital PCR-based kit. Just as a little background and continuation from what Sarah had mentioned earlier about this 20q abnormality, and it's probably the same publications that she cited earlier. This 20q gain copy number variant is detected in well over 20% of the worldwide cultured stem cells at this point. It is by far the most common genomic abnormality that's present in about 23% of the recurrent structural variants that are identified in stem cells. And as Sarah had mentioned earlier, these cells often harbor a selective advantage. It can outgrow the wild-type cells and overtake the culture within a few passage. And to add to all of that, most of these 20q mutations actually fall outside the detection limits of many of the conventional, methodologies for testing this, such as G-banding or even the microarray-based technology that, such as Karostat Plus. So when we found this out, EditCo as a team internally wanted to investigate this and tested our earliest passages of this parental repository that we had using this PCR technology that IDEXX employs. And what you see here using the traditional microarray-based technologies, the genomic abnormality was not detected. However, using this ICS digital PCR method that IDEXX employs and offers, we're able to detect this 20q abnormality in our basic working bank of PGP1 cells. Since this discovery, though not all is lost, we were able to clonally isolate a genomically stable PGP1 cell line to recreate our master banks. But I think this is a prime example of how cell health and genomic stability plays an important role in research. As previously stated, this 20q abnormality allows for the selective growth advantage compared to wild-type cells, and if left unchecked, it can lead to providing false positives in downstream assays, unbeknownst to researchers such as yourself, if they aren't properly checked out ahead of time. All that to say is I also want to note that many commercially available iPSC lines are a bit genomically unstable when analyzed via traditional methodologies such as microarray or G-banding. So we worked with, as I said, many cell lines on our platform, and we wanted to go back and look at some of these other commercially available cell lines that we previously worked with that are listed in the chart on the slide there. I've removed what these actual cell lines are because I would prefer to keep the actual names of these cell lines from being publicized, but We do see that there are several of these commercially available cell lines that if you use traditional methodology, you would not detect any of these abnormalities. However, with the ddPCR technology that IDEXX employs, you are able to detect these sort of abnormalities that are important for cell health and the stability and of the cell line. So it's important to continually detect these cells, whether you get them commercially or they're your in-house lines with IDEXX and their service provider. So in closing, I just want to highlight again the importance that we take cell health at EditCo and why we find IDEXX to be such a great partner and to make sure that we can maintain the highest quality and reproducibility we see on our platform. While I briefly talked about the genomic stability, one of the other ways and very important aspects I didn't touch on in detail is the cell qualification check that IDEXX provides us for all of our customer supplied cell lines. Before we even start culturing cells from customer supplied cell lines, we wait on the seal of approval from IDEXX and with regards to sterility and pathogen testing. All customer supplied cells that are sent to EditCo must have an AH impact testing performed before we bring them into our facility to work with them. It's important for us to maintain the cleanest of environments, and IDEXX provides us really with that assurance that we need, not only protecting other customer provided cells in our internal cell lines, but also our researchers because of the pathogens and whatnot that are tested for by IDEXX. So one last slide from me, and that really is just to highlight the partnership sort of ecosystem that I like to say that we have with our IDEXX partners. So IDEXX, as you said, is the top right there. Really, we started a lot of our projects with this H impact testing or genomic stability testing or any of the other services that they're able to provide. Then we can get those cells. We can provide any sort of genomic editing solution that our researchers want to bring to us. But on the backside of that, for all of our projects, we check genomic stability on the front end, and we offer the service to check the genomic stability on the back end. And so that's where we feed back into that ecosystem and give our cells back to IDEXX to check out and make sure they're still healthy on the back end as well. All circling back to researchers like yourself to make sure that you have the highest quality projects and reagents available to you to perform your research. And then back in the ecosystem again, if you want to make sure that cell health is maintained over time. So with that, I believe I am done. I will pass it back off to Steve to see if we've had any great questions come up.
Awesome. Thank you very much, Travis and Sarah. Terrific stuff. Really enjoyed listening to all of that. And yeah, we have some thank you guys. Thank the audience very much. We've had some really nice questions come in as well. So let me get through a couple here. So Travis, since you were up recently here, let me start with you. There's a question. With 800 cell lines through your facility, what are the trending applications or research areas you see for gene editing services today? What are the hot buttons you see coming in?
Yeah, I mean, obviously we can see a lot of and pick up a lot on, the neuro is a big sort of hot topic with iPSCs, particularly we're talking about iPSCs, before. So we see a lot of trends with customers who are interested in specific, the hottest sort of mutation for Parkinson's or Huntington's disease when it comes to an iPSC cell line. So, through different partners, we have differentiation services. And so we can create a specific neuronal, disease-associated mutation and then, provide that to facilities that can actually differentiate those into neurons for researchers. And so that's sort of a hot. We always, neurological based research is always hot. And then, any sort of drug discovery, early phase, screening of different sort of targets for drug assay or drug discovery and things like that.
Okay, cool. Thank you. We'll go back and forth. Sarah, this one I think is for you. So regarding cell health, what should I be looking for in the lab? The question is, can you give any examples of issues that have come up in cell research from contaminations? So practical, or maybe do you have any anonymized examples of what people should be looking for in labs?
Yeah, I think issues in the lab. So actually, we have another document that we share sometimes, and it's kind of a good cell handling type of protocol, basically, and you can kind of follow through it and look for where there may be issues happening in the lab. And sometimes it depends upon the lab's experience and the individual's experience on what they're doing. But for example, when we see evidence of contamination, So cell contamination, whether that is from the same species or from a different species, one of those steps is going back and checking to see how are you handling your cells? Are you opening multiple vials at the same time? How are you cleaning in between, et cetera? And so kind of going back to some good cell handling techniques is usually the first thing. And then one of the things that we see, so we get a lot of questions about our mycoplasma assay, and individuals will ask, what type of assay do we do? Are we culturing these? And so we do run real-time PCR for our mycoplasma testing. And so mycoplasma is something that can, when you get it into your cell lines, it can take over very rapidly. So routine screening by an outside lab for mycoplasma is something that I think is really important as well. But yeah, a lot of things from the lab, and many times what we can do too is we can work with investigators when we do see issues with misidentification or contamination, and we'll go back kind of step by step to previous times that those cells have been banked to look and see where that may have occurred. And kind of along those lines, there's so many different things that we work through and things that we find, but labeling is actually still a big one. So ensuring that the labels that are on the cryo vials are appropriate, easy to read, et cetera, because we do still see issues of misidentification just due to labeling issues.
Can I add something to this?
Always a good one, right? Always the label gets you.
Yeah.
Steve, can I add something to that real quick? I just want to say another testimony to, I think, the IDEXX and IDEXX sort of methodology team in general, right? I can't tell you how many times that we've received cells from customers. We've sent them to IDEXX to have tested for mycoplasma, and they've come back as dirty, right? As having a little bit of mycoplasma contamination. And it's interesting that we will go back and talk to our customers, like, how's that possible? We test, or we look in our, we use our, whether a colorimetric-based assay or whatnot, and the cells are perfectly fine. I don't know how this is possible. And I think it goes to show that the sensitivity that the IDEXX team is able to pick up, far sooner than a lot of the other methods out there is really important. And we find it extremely beneficial, where we have never had any sort of outbreak in our labs because of them. We trust you guys for that. So another testament to you guys.
Yeah, thanks. Appreciate that. And as you're getting into these cellular models, the sensitive detection really goes a long way. Some of those kits don't cut it. Here's a good one. Here's one for both of you. This is a fun question. Thank you, Nahid. Excellent question here. And Sarah and Travis, you guys can kind of fight over this one. Nahid says, I need a human iPSC line which can be differentiated easily into cardiomyocytes. Looking for reliable detection of mycoplasma in human PSCs as well. So can you test mycoplasma across stem cells, and an HPSC line can be differentiated into cardiac myocytes? That's a question for both of you. Nice job. So
I'll let you go first.
Okay, I'll go first. So for the kits for detection of mycoplasma, so since we don't develop or run any of those kits, I can't speak to their function or their sensitivity. So I can only speak to ours that we run here. So we do run, like I said, real-time PCR. Our turnaround time is 3 days or less for your results on that. And typically, so our PCR assays are validated to a level of 10 copies. So if you're trying to decide if you're going to do something in-house, then I would be looking for that level of sensitivity, because I think that's what you need when you're trying to keep mycoplasma out.
Great.
And Steve, can you repeat the second half of the question? That was
an iPSC line differentiated into cardiomyocytes. How feasible is that?
Yeah, that's actually really feasible. This is something we have quite a bit. So cardiomyocytes and any sort of neuronal differentiation is a lot of the common sort of downstream pathways that we see a lot of researchers take with the edits that we make. And so, yeah, our PGP1 cell line, there's a lot of publications out there, and I can provide that list, I think, afterwards if you would like, of customers who've taken that cell line and actually differentiated them into cardiomyocytes and published some great research with it. And so can easily be done. some of the other cell lines we have on platform have also been differentiated in the cardiomyocytes. And so, yeah, it seems to be something that is relatively easy to do at this point.
Okay, perfect. Well, that's a good one to end on. Really appreciate the question, Nahid, and shows the reason we're doing this today. There's all kinds of super cool techniques and advanced cellular models that have come about by CRISPR editing that the EditCo bio can help with and consult with and deliver and help you decide what you need and how to make it. And then all along the way, before editing and while they're in use in your lab, the importance of testing for mycoplasma and cell line authentication, tracking your cells as cells have become the rule rather than the exception in as the foundation of research programs. And glad to see people paying attention and interested in doing good QC on their cell work along the way. So a reason why we love working with EditCo and learning what they're doing. And so hopefully you all did as well today. Thank you for joining us. This recording will be sent out for anyone who couldn't join us, and feel free to share with your colleagues and reach out to myself or Sarah or Travis with any questions. Thanks very much.